The third category of pigment molecules are the porphyrins. This is going to include chlorophyll, it's going to include cytochromes, and it's going to include a big focus on hemoglobin and myoglobin. So porphyrins are a group of pigments that have lots of different functions in plants and animals, but we're going to look at those ones I just mentioned. What they all have in common is a planar, so a flat ring structure. Within a larger ring structure are 4 heterocyclic rings. A heterocyclic ring is a ring that contains an element other than carbon. In this case, the other element is nitrogen. And there are some other bridging carbon atoms between them. The rings are going to act as Tetra dentate ligands. We could also just call them polydentate ligands, but tetradentate meaning 4 teeth ligands. So this giant structure, this sort of planar ring structure, is going to donate 4 electron pairs to a central metal ion or metal atom. So the ring itself is acting like one giant ligand that actually gives 4 dative covalent bonds to the central metal particle, whether that be an atom or an ion. So that's why they're called tetradentate ligands. They can also be called chelates because of the number of dative covalent bonds they're forming with the ligand. So each nitrogen atom on the ring forms 1 dative covalent bond with the metal, and because there are 4 nitrogen atoms, they end up forming 4 covalent bonds with the metal. Porphyrin groups can have different metals at their center. They can also have different groups sticking off to the side. Just to start it off, here's an image of chlorophyll, where the R group here can change and be chlorophyll A or B, but you'll see that these are the 4 nitrogens, and you can see them forming these 4 dative covalent bonds with the magnesium atom in the center. And then over here, we've got the heme group of hemoglobin and myoglobin. Again, the porphyrin structure being the rings here. You've got these, there's 4 rings that look just like that if you look around the structure. So those are the 4 rings, the heterocyclic rings, and then they are connected by these carbon atoms here. Each ring gets connected by carbon atoms, but you'll see that there's high levels of conjugation all the way around the outside of these rings. There's alternating double and single carbon-to-carbon bonds, or carbon to nitrogen bonds. You also have metal ions, and so while magnesium doesn't contribute anything to the color, something like iron that's a transition metal can contribute to the color of the porphyrin pigment. So they all have that structure in common, that planar 4 heterocyclic ring structure, but not all of them have a metal ion that contributes color to them. And I'm playing with the zoom, sorry about that. Okay, so it's this high level of conjugation, and then what I just said, some of them, the central iron ion, can also contribute. So first we'll look at cytochromes. You actually don't need to know anything about the color of cytochromes. It's a little bit odd that they include it in the pigment topic, but they are important porphyrins, so they decided to include it, I suppose. So cytochromes act as electron transport carriers, which is part of the respiration process, involves transporting electrons. The iron ions in the cytochromes can both be reduced and later oxidized. So when they gain electrons, they are reduced to the iron 2 form, and later they get oxidized to the iron 3 form, and they pass the electrons on to subsequent electron carriers. The chain of carriers, each sort of porphyrin or each carrier contains different metals, and it's actually has evolved to contain different metals, each of which has more and more affinity for electrons. So if you think back to the redox topic, we would say each metal has a more and more positive electrode potential. It's more and more spontaneous for the metal to accept electrons. So for example, if iron is higher in the electrode series than copper, there is a cytochrome further down the electron transport chain that will contain copper ions. And so as the iron has the electrons naturally will spontaneously move to the cytochrome that has copper, because that's how the electrons are more spontaneously going to transfer. They're more attracted to the copper ions. So you don't have to know anything about other cytochromes. You only have to know this example that contains iron. But I just thought I would fill that in. The The last cytochrome in this chain donates electrons to oxygen, which is what causes oxygen to get reduced to water. So again, this is in respiration. So this is in the process of getting energy out of glucose. And so cytochromes are important electron transport carriers in that process. Cyanide ions bind irreversibly to the metal ion in the final cytochrome in the electron transport chain. And so effectively they prevent cellular respiration from completing. And that's how cyanide acts as a poison. The next porphyrin is chlorophyll. Chlorophyll, there's two different kinds of chlorophyll, and they absorb different parts of the visible spectrum, but they combined absorb a lot of red and blue light, which results in them making things appear green. So other accessory pigments, the carotenoids, can absorb light and pass energy to chlorophyll, in addition to the red and blue light that chlorophyll absorbs. When enough energy is absorbed by chlorophyll, it undergoes a redox change. It gives electrons to other molecules, which are those electron transport carriers. And these electrons eventually will take part in the reduction of the carbon dioxide and turn it into the glucose, into the carbohydrate. The chlorophyll itself can later be reduced back to the original state, producing oxygen, or by water, which produces oxygen. So part of the reason why oxygen is a byproduct is that water is actually getting oxidized into oxygen and donating its electrons back to chlorophyll to convert it back to the original kind of chlorophyll. So you've got the light coming in, which excites, the photon causes electrons to be excited and lost from the chlorophyll, and that's what kicks off the process of transferring electrons down a chain. There are some factors that cause changes in the color of chlorophyll. If there's magnesium deficient soil, that can lead to a lack of chlorophyll because you need a magnesium atom in the center there. And that results in plants appearing less green and more yellow because those carotenoids that are beneath the green or in less concentration than the chlorophyll can then shine through. If you decrease the pH of a chlorophyll environment, then that can cause the magnesium to be replaced by H+ ions, and that causes a change from a bright green color to sort of a darker olive brown color. If you've ever done some cooking and you take a plant of some sort like spinach or kale and you sort of bust up, you massage the kale, as they say. If you break up the cell walls in plants, they often start to darken because as you break up the cell walls, that often releases small amounts of acid that are within the plant. And so those acids are causing this same color change within the chlorophyll. The H+ ions are replacing some of the magnesium ions, which causes them to look darker. If you cook plants in your vegetables in slightly alkaline water, like by adding baking soda, apparently it can keep the vegetables looking brighter green because it prevents this change from happening. The last pigment, and the one that gets the most focus, is heme. So heme is a group. It's called a prosthetic group because it's this structure, and then it actually is attached to a protein chain, a peptide chain within hemoglobin and myoglobin. It contains iron in the +2 oxidation state, and heme shows up in both hemoglobin and myoglobin. So first of all, what are those two things? Hemoglobin is the protein that is responsible for absorbing oxygen in the lungs and transporting it throughout the blood and throughout the body. Hemoglobin has 4 separate peptide chains. So it has this quaternary structure that we've talked about before. Each one of those peptide chains has a heme group, which means that hemoglobin can carry up to 4 oxygen molecules at a time. Myoglobin is similar, but it's only one peptide chain. And so it only has a tertiary structure and it only has one heme group. Myoglobin is the protein that's responsible for storing and eventually releasing oxygen in the muscles. So oxygen molecules bind reversibly with the iron 2 in the heme group. So it can bind but also be released. And each heme group can bind to 1 oxygen at a time. So as I said before, hemoglobin can bind 4 and myoglobin 1. It does not cause a change in oxidation state, so they don't say that the iron is oxidized, they say it's oxygenated. It's pretty simple, Le Chatelier's principle, to explain how it works. In environments where there's high concentrations of oxygen, the oxygen is more likely to bind up to the hemoglobin and form oxyhemoglobin, where it's bound reversibly to the hemoglobin. If you enter an area where there's a low concentration of oxygen, then it's more likely to be released to the cells that need it. Same thing goes for myoglobin. If there's lots of oxygen around, it will bind the oxygen, and when oxygen is used up and there isn't oxygen in the muscle cells anymore, then it will release the oxygen to the muscle cells. So Le Chatelier's principle is a very simple way to think about how the oxygen would know to bind or to be released. It doesn't really have to know anything. It's just what's going to happen. So when you're in an environment like the lungs, where there's a high oxygen concentration, that's going to cause shifts to the right in these equilibria, which means that those proteins are going to bind up with lots of oxygen. And if you move out into the body where you're at a place where respiration has been taking place and the oxygen has been depleted, then the oxygen will naturally be released. And this is just a little graph showing absorbance, and it's showing that there are different wavelengths of light that are going to be absorbed when hemoglobin has oxygen bound to it or not, and that changes the color of blood depending on if it's oxyhemoglobin or deoxygenated hemoglobin. So in myoglobin, there's only one heme group to work about. So the binding of the oxygen shows a normal enzyme standard saturation shape curve where it goes up quickly at first and then eventually flattens out when the myoglobin is totally saturated with oxygen. As the oxygen increases, it gets increasingly saturated. So when we talk about oxygen concentration, we're going to be using this term partial pressure of oxygen. In hemoglobin, however, it's a little more complicated. The binding of oxygen is called cooperative, which means that as each oxygen binds to the hemoglobin, to the heme group, it causes little changes in the shape of the protein, which actually make hemoglobin better at binding to oxygen. So the more oxygen it has, the better and better it gets at binding to the oxygen. And so this is trying to show that as it binds to the oxygen, it sort of flattens out the heme group, which triggers changes in the shape of the entire hemoglobin molecule, which changes the affinity for oxygen. So what that means is that while myoglobin is going to look something like this, where it goes up with increasing oxygen concentration and then eventually hits a saturation point where it can't absorb any more oxygen, hemoglobin has what we call an S or sigmoidal curve shape. So it has a shape sort of like this. So it starts off slower, then it goes up steeper, and then it's going to eventually plateau out. Now, it almost is like a titration curve where it's slow at first, then it's faster, 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 and then it's fully saturated and it can't really take any more oxygen. So this is hemoglobin, and the top one there is the myoglobin. Okay? So a couple things. What this shows is that myoglobin has a stronger affinity for oxygen than hemoglobin does. That's really important. That means that if hemoglobin is near myoglobin, the oxygen will naturally move from the hemoglobin to the myoglobin, the thing that has a greater affinity for it. So that means that you can take hemoglobin that has oxygen, and as it passes by the muscles, it will naturally transfer oxygen to the muscles. It also makes myoglobin a much better molecule for oxygen storage because it holds on to the oxygen fairly strongly. So it's not very good at transporting it because it wouldn't release it very easily. So myoglobin has a stronger affinity for oxygen, and that's a useful thing. There are some factors that decrease the affinity of hemoglobin for oxygen, and that's actually really good and important for hemoglobin to carry out its function. One is increasing temperature. So if hemoglobin passes into a part of the body where the temperature is a little bit higher, it's going to make the hemoglobin release the oxygen more readily. Now, that's really important because if your cells have been undergoing a lot of respiration, they're more likely to have caused the temperature in that area to be higher. And so hemoglobin is naturally going to release oxygen in the areas that need it most, that have just been doing a lot of respiration. The other one is decreasing pH or increasing CO2 concentration. Again, cellular respiration means more CO2 is produced, which leads to more carbonic acid being produced, which lowers the pH. So these two things go hand in hand. Again, hemoglobin doesn't bind to oxygen as well when it's in a place with more CO2 and lower pH. And so that means that when hemoglobin passes near cells that have been undergoing respiration, it will more likely release its oxygen. So if this is normal body temperature, like 37, if the hemoglobin is passing by something that's warmer, like 40, it has less. Here's hemoglobin's affinity at 40. So it's less affinity, less saturation with oxygen, which implies that it's going to release, this hotter one is more likely to release the oxygen than the one that's cooler. You'd see a similar pattern for the pH. So a higher pH would be something like this. And then here's your lower pH, again, less affinity at lower pH. So it's more likely to release its oxygen in places that are warmer, in places that are lower pH, and both of those go along with lots of cellular respiration. The last thing about hemoglobin is that there is a different kind of hemoglobin that is in the human fetus. So it's called fetal hemoglobin. It has different chains. Instead of 2 alpha and 2 beta peptide chains, as they are called in hemoglobin, it has 2 alpha and 2 gamma chains. You don't need to know anything about those structures, just that it's different and that it has a greater affinity for oxygen than adult hemoglobin. Again, that's really important because if adult hemoglobin is passing by the fetal hemoglobin, the oxygen will naturally transfer from the adult hemoglobin to the the fetus's hemoglobin, and thereby it can provide oxygen for the fetus before the fetus can breathe. So if we draw curves, once again, if this is mama, then baby looks like this. And so this is the fetal one, and the other one is the adult one. And so the oxygen is naturally going to transfer to the fetal one because it binds it stronger. And the myoglobin would be higher than both of them, and looking like that normal enzyme saturation curve. So the last couple of things, that's a space to summarize the things that we've talked about. So we've talked about increased affinity would be myoglobin, and then fetal over regular hemoglobin. We've talked about how high O2 concentration leads to greater affinity, and it's actually cooperative in hemoglobin, binding more to it. Decreased affinity is regular hemoglobin, but also higher temperatures and lower pHs / greater CO2 concentrations are things that cause also low O2. And these are all things that help hemoglobin lose its oxygen where it's needed most. Last couple of factors to realize are carbon monoxide is a poison because it irreversibly binds with iron 2 in hemoglobin. It has a 200 times greater affinity for the iron than the oxygen does. And so it renders that heme group unable to carry oxygen. So if you get enough CO into your body, into your blood, it will render all of your hemoglobin useless and you can't carry oxygen around your body and you will die. So it effectively acts as a competitive inhibitor to that protein. Even though we don't really think of, let me write this. We don't really think of hemoglobin as an enzyme. The carbon monoxide effectively acts as a competitive inhibitor that irreversibly binds to the iron so that it can no longer carry out its function of carrying around the oxygen. Last thing is that cooking, you may have noticed, causes changes in color, as does leaving meat out causes changes in color. And that's because the iron 2 can get oxidized to iron 3 when you cook it or when you leave it out and expose it to oxygen. And so the IV focuses on the cooking, but the cooking causes the color change from red to brown of that meat. And that has to do with how that metal ion is changing oxidation states, which changes the energy wavelengths that it absorbs from the visible light. And so that is the topic of porphyrins, looking at cytochromes, chlorophyll, and especially at the functioning of hemoglobin.